Article 7, which is bone and ossification. This is Dr. Byson, EM. Without further ado, let's get into this lab and see what is expected of us. Let's have a little bit of introduction even before we look at the histological slides. Bone is a specialized connective tissue. This you must know. And there are two types of bone tissue, which are the compact bone and spongy bone. So you have compact bone and spongy bone. So the two names imply that the two types differ in density and how tightly the tissue is packed together. So as you are going to see, compact bone is tightly packed together and spongy bone just has trabeculae. It's not tightly packed together. There are three types of cells that contribute to the well-being of bone, or a normal bone, which is bone homeostasis, which are the osteoblasts. So the osteoblasts are bone-forming cells, and there are osteoclasts, which resorb or break down bone, and osteocytes, which are mature bone cells. So when your osteoblasts are embedded in bone matrix and become mature, they are known as your osteocytes. Now, an equilibrium between your osteoblasts and osteocytes maintains bone tissue. All right. Now, what are the objectives for this lab? So at the end of this lecture, students should be able to, number one, describe the different types of bones and their general characteristics, describe the cell types found in bones and their functions, describe the light microscopic features of the compound and spongy bone as well as the components of the bone matrix. You discuss the process of intramembranous and endochondral ossification, identify and describe different zones of epiphyseal growth plates, and lastly, you discuss the process of bone repair. So these objectives are very important because these are the same objectives that we bring in theory histological questions in anatomy. So once you clearly explain and understand this, then the histology practical and the theory won't be a problem for you. All right, now let's look at compact bone, which is from slide number 6 from the rib. Okay, so now you survey the whole section of the slide that I'm going to show you and note the organization of the bone into peripheral compact bone and the central cancellous bone. So you sketch this, and in compact bone you identify the Haversian systems, each consisting of concentric lamellae enclosing a central canal and demarcated peripheral by a central line. You note the outer and inner circumferential as well as the intermediate lamellae. At high power, you identify the canaliculi. The canaliculi, what do they contain? So you examine individual Haversian canals at high power and note the structures that contain. Now the task is to only draw the Haversian systems at high power. Draw the Haversian systems at high power. All right, let's look at the slides. So this is the slide. So this is cancellous and compact bone. So compact bone is this bone here that you find in the periphery, the sides. So this is compact bone. And then what you see inside here, this is what we call trabecular. So this is cancellous bone or trabecular bone or spongy bone. So this is compact bone. You can't appreciate the Haversian systems that are here at low power. And then this is cancellous bone. Let's look at this at high power. So high power, this is medium power actually. So it brings into view, these are the Haversian systems. So this is the Haversian system, this is the Haversian system, this is the Haversian system. This is compact bone. This inside here is called the trabecular. Okay, so this is trabecular bone. Trabecular bone has got these white and purplish structures. This is the bone marrow, and then this is the trabecula. The trabecula is what contains the Haversian systems and the cells that make up bone. This is the bone matrix in the cancerous bone. All right, now let's look at the Haversian systems at high power. So this is the magnified compact bone. So in the compact bone, as you can see, this is the central canal. This circle here is what we call the Haversian system. The Haversian system is the functional unit of a bone. It is also known as an osteon. It is also known as an osteon. So in the osteon, you have on the middle here the central canal. The central canal, and then you have concentric rings here. These concentric rings are known as the Haversian lamellae. They're called lamellae. Lamellae. Now, in the lamellae, as you can see, let's look at this. This is the central canal. In the lamellae, you have this, what you see here, what you see here, what you see here. These are called lacuna. Now, the lacuna have got cells in there that are called osteocytes. The osteocytes are the mature osteoblasts. So after the osteoblasts deposit bone matrix, which is called osteoid, and the osteoid is calcified, they mature into osteocytes. So what you find in the lamellae is lacuna. The lacunae are the spaces which contain cells known as osteocytes. Okay, so this is high power Haversian system. So draw the central canal here, and then you show the lamellae, and then in the lamellae you show the lacuna. Inside the lacuna, then you show the osteocytes. Okay, so if this diagram is not very clear for you, I have put another diagram here so that you can see it. Okay, so this is a schematic diagram of an osteoid, an osteon. Okay, so this is an osteon. You've got your central canal here. This concentric ring is what we call the lamellae. These concentric rings are the lamellae. So as the lamellae are concentric, you can see this here is what we call the lacunae. The lacunae has got a dark cell inside here. That is what we call the That's what we call the osteocyte. Okay. Now, if you look at this one here, it shows the Volkmann's canal. The Volkmann's canal is simply a canal that communicates arteries and veins and the lymphatics from the blood vessels that are running in the connective tissue that surrounds the bone called the periosteum. So the periosteum is going to contain blood vessels, and then those blood vessels are going to penetrate the Haversian system through what we call the Volkmann's canal, so that there's going to be blood supply bringing nutrients and oxygen to the bone tissue. All right. So at high power, so this is low power compact bone. Okay, now this compact bone, the connective tissue and the excess cells have been removed so that the Haversian systems can be shown very clearly. When you remove the connective tissue and the cells so that the Haversian systems can be seen very clearly, this bone is known as ground bone. So compact bone that has got very clear Haversian systems where the cells and the connective tissue has been removed is what we call ground bone. So low power ground bone. Let me show you a high power ground bone that shows a very clear Haversian system. So this is ground bone showing the Haversian system. This is the central canal here. And then these concentric rings are what we call lamella. This is a lamella here. This is a lamella here. Now in the lamella, you can see this is a lacunae that contains an osteocyte. Contains an osteocyte. So it's very easy to draw the osteon, which is a functional unit of a bone, the Haversian system in ground bone. So in ground bone, just come and draw the Haversian system. So label and show the central canal, the concentric lamellae, the lacunae, and the osteocytes. Now, you can see these projections. So from this lacunae, there are these projections that are coming like that. These projections that are appearing like lines here. They appear in like lines here. These are communications between this lacunae and this lacunae. So you must know what are these. What do you call these lines? Identify them and label them as well. So these lines are what we call canaliculi. They're called canaliculi. They're just channels of communication from the central canal to the lacunae. Yeah. So we've already looked at this already. So this is a compact and cancerous bone at low power. All right, so this is a cancerous bone at high power. So we took it at high power. This is what we call the trabecula. Trabecula. And what you can see here is the bone marrow. So the bone that we were looking at previously was compact bone. This is cancerous bone. So cancerous bone, when you're drawing cancerous bone at high power, you're only drawing it at high power. Show the trabeculae. When you see the trabeculae, when you show the trabecula, then show the bone marrow. Okay, so these are adipocytes. The white cells here that are appearing white are adipocytes. And then this is the bone marrow, which has got hematopoietic stem cells that produce blood cells. So as you can see in the trabecula here, you can see this cell here, 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 this cell here. These cells are in spaces called lacunae. So there's a lacunae, and inside of the lacuna, you've got cells that are known as osteocytes. So draw this. These are collagen fibers, and then draw the lacunae. From the lacunae, show that inside the lacunae there are osteocytes. In the peripheral spaces here, we've got cells, although they're not visible, that are known as osteoclasts. We've got cells that are known as osteoclasts. So draw this slide, which is a cancerous bone at high power only. All right. Now, we looked at the two types of bones histologically. We looked at the the compact bone, and we showed the Haversian systems, and we looked at the cancerous bone and showed the bone marrow and how the lacunae are arranged in the trabeculae. Now let's look at the two types of ossification that are present. So in human beings, for bone formation, there are two types of ossifications. There is intramembranous ossification, which is an ossification that we use to make the bones of the calvarium, the skull, and the clavicle, intramembranous calcification, and also the tooth. And we also have another type of bone formation, which is called endochondral ossification. So let's start by looking at intramembranous ossification. So intramembranous ossification is a type of ossification by introduction where bone is formed inside of your mesenchymal tissue. Mesenchymal tissue. So bone is formed inside of the membranes. That's why it's called intramembranous ossification or calcification. Intra means inside. Membranes is just tissue. So intramembranous calcification or ossification. So what happens is that you have a tissue, and inside of those tissues, cells inside of the tissues differentiate into osteoblasts, and your osteoblasts begin to produce osteoid. The osteoid is calcified. Okay, so this is low power. This is a tissue. This is embryogenic tissue. So this tissue is known as mesenchymal tissue. Now, this mesenchymal tissue has got what we call mesenchymal cells. The mesenchymal cells are going to differentiate into osteoblasts and osteoclasts. And then the osteoblasts are going to start forming bone tissue that is called osteoid. As you can see, this is the mesenchymal tissue, and on the middle here there is bone tissue that is forming. This matrix of bone that is deposited here is called the osteoid. Osteoid would then now have deposited calcium to form bone tissue. So inside of the membranes, the mesenchymal cells are going to differentiate into osteoblasts. The osteoblasts will then deposit bone matrix that is called osteoid. Osteoid will then be calcified and bone tissue will start forming inside of the membranes. So this is what we call intramembranous ossification. As you can see, there was just membranes here, and inside of the membrane bone is forming. That's how a tooth develops, by intramembranous ossification. Other bones that develop by intramembranous ossification, the bones of the skull and the clavicle, the membranous part of the clavicle. Let's look at it at high power. So at high power, this is what happens. Listen, so this is the mesenchymal tissue. The mesenchymal tissue has got cells that are called mesenchymal cells. The mesenchymal cells are going to differentiate into osteoblasts. Osteoblasts will start depositing this tissue here that is called osteoid. Osteoid will now start getting calcified. So bone tissue is formed inside of the membranes. That's why this is called intramembranous calcification. Okay, so what you see, this here, this here, this here, these are just lacunae, and the lacunae have got cells that are called osteocytes. So when you are drawing this high power, label this as in mesenchymal tissue, and then these are lacunae containing osteocytes. This is osteoid being calcified. These cells here that you see, these cells here, these are osteo crusts. So this is osteoclast, osteoclast. And then these cells in the middle that are small are osteoblasts. So this is all about intramembranous ossification or calcification that happens in a developing tooth. So in your histological examination or practical exam, if they ask you what slide this is a developing tooth that is showing intramembranous calcification. It's very easy to identify it because it's the only slide that appears green in histology at UNZA. Okay, let's move on to endochondral ossification. So this is just high power intramembranous calcification, and it's showing how your bone tissue is forming inside of your membranes. Endochondral ossification. So endochondral ossification is a type of bone formation where the bone is formed from hyaline cartilage. So hyaline cartilage becomes calcified and it forms bone. So all the bones in the body apart from the bones of the skull and the clavicle formed by endochondral calcification. And the cartilage in fetuses or in babies will become bones by a process of endochondral calcification. So let's look at how this one happens. Right. So we're looking at it from the finger. So what will happen is this. What you see here is a layer of hyaline cartilage. The hyaline cartilage will undergo processes and it will become bone. The processes is that hyaline cartilage, first of all, is reserve cartilage. It's in a resting zone. Now the resting zone, the cartilage will start proliferating. After the cartilage proliferates, it will hypertrophy. After it hypertrophies, then the hypertrophied cartilage is going to be calcified. After calcification happens, then bone is going to form. So as you can see, this is cartilage here. This cartilage undergoes processes and it becomes bone. So this is trabecular bone here, and then this is spongy bone. So from cartilage, you're able to see this is a layer of cartilage. From cartilage, trabecular bone is formed and compact bone is formed. So for this, first of all, draw at low power. So you draw this at low power, and then after drawing at low power, you go to high power. low power. Then this is high power. So this is high power. So this is cartilage in the resting zone. So this cartilage is resting, and then after the cartilage rests, it will start proliferating. So you can see here there is a lot of cartilage. How do you know that this is hyaline cartilage? Because this appears like a glass-like appearance, where you have lacunae, and inside of the lacunae, you've got your cartilage cells that are called chondrocytes. Chondrocytes. Okay. Hyaline cartilage, remember, has got type 2 collagen fibers. So that's type 2 collagen fibers. So the chondrocytes that are in lacunae here will start proliferating. They'll start increasing. After they proliferate, then they will become big, meaning they are hypertrophied. After they are hypertrophied, then they will be calcified. After calcification, then bone is formed from cartilage. That's what we call endochondral bone formation. So now this schematic diagram here just shows you the layers of bone formation in endochondral ossification. So this layer here is reserve cartilage, resting cartilage. After the resting cartilage begins to proliferate, meaning that it becomes a lot. Have you seen here? Then after it proliferates, it hypertrophies. After it hypertrophies, then calcium is deposited in the cartilage. So this is the zone of calcification. After calcification happens, then bone is formed from cartilage. So this is endochondral ossification. Very easy and very nice. I hope you guys are enjoying this as much as I am. Alright, so those are the types of bone formation in the body. There are two: intramembranous ossification and endochondral ossification. Now let's look at the epiphyseal plate. The epiphyseal plate is what is referred to as the growth plate. So you may ask, how does bone grow? So bone grows because there is a layer of cartilage in long bones that is maintained, and this layer of cartilage this layer of cartilage, this layer of cartilage proliferates, and as it is proliferating, new bone is forming from that layer, and that's how bones grow. So if you have a long bone, you've got a shaft that is known as the diaphysis, and the head that is called the epiphysis. So at the epiphysis, there is a layer of cartilage. That layer of cartilage is responsible for bone growth. The layer of cartilage that is responsible for bone growth is what we call an epiphyseal disc. Okay. So now we're going to look at layers of the epiphyseal disc. We have the resting zone, the proliferative zone, the hypertrophic zone, ossification zone, and the ossification zone, which has osteoblasts. Now let's look at the histological slide from the femur, which is slide 8 that you're going to draw. All right. So we're going to look at that slide, but after this slide that I'm going to show you, you're going to do this exercise, which is you draw the cells of the bone and state their functions. You contrast woven bone from lamellar bone. You draw a well-labeled diagram to illustrate a synovial joint. You discuss in detail the zones of endochondral ossification, something that we've already looked at. You explain the causes of the following conditions: osteoporosis, osteopetrosis, osteogenesis imperfecta, osteomyelitis, and acromegaly. And you highlight the difference between rickets and osteomalacia. 7, you explain the phases of fracture. Okay, so that's the exercise that you are doing. Now, these are the zones of the epiphyseal plate. So you're going to draw this, the zones of the epiphyseal plate. So you start with this one here. So this is the epiphysis, where the bone grows from. So this is the layer of cartilage, which is the reserve zone or the resting zone. The resting zone will start proliferating. So this is the proliferation zone. As you can see, cartilage is proliferating. After it proliferates, it hypertrophies. Hypertrophy is to grow in size. it will hypertrophy. After it hypertrophies, the hypertrophic zone, it will now be calcified. So this is a zone of provisional calcification. After it calcifies, then it forms bone, which is the metaphysis. The metaphysis is the junction between the diaphysis, which is down here, and the epiphysis. All right. So what you also have to know is that diaphysis, in the diaphysis, there is a region of primary ossification. What that means is that that is the first place in the bone where cartilage is turned into bone. And then the epiphysis has got secondary ossification centers, and it takes a long time for it to totally ossify. That's why for the bones to be totally ossified and for you to stop growing takes about an average of 21 years in the human being. All right, so that is the slide that you're going to draw for the epiphyseal plate. To study further, you go to Janquilla chapter 8 and study bones, bone formation, and bone repair. This is Dr. Byson EM, creating histological slides and all the topics in med school. Don't forget to comment, to like, to subscribe, and to invite your friends to this channel. I'll see you in the next video.